Okay, while people are filing in, we'll get started so that we can stay on time. Welcome to Sunday morning. I'm the chair of this session. My name is Erin Rich. The session is called Neural Coding and Representations. And I'm pleased to introduce our invited speaker for the session. Max Shine is at the University of Sydney, and his title is The Role of the Thalamus in Complex Adaptive Dynamic Behavior. Take it away.
All right. Everyone hear me okay? Yes? All right. Hi, everyone. It's an absolute pleasure to be here today at a conference that I've only been attending for a couple of years, but is an absolute inspiration to the work that my group and I have been conducting over the last few years. I come from a land quite far away from here in the Southern Hemisphere, Australia. This is the university that I work at here, the University of Sydney, in this little dot. And one of the main questions that we get asked when we come to conferences so far away from where we live is what it's like to be so jet-lagged. We're often quite tired. I think it's something like about 11 p.m. now in Australia, but that's great, right? We get to come to a conference and we come with jet-lag brain too. So that means we get to come with this nice kind of loose set of associations, and we can meet really amazing people and try and learn about how we can update our work. And so I want to start today to talk about some of the work that we've been doing. I come from a different community as well, from the human brain imaging community. And I want to start with a motivating example of what it's like to come from that community to a community like Cosyne. So here's the poster hall at SFN. It's very similar to a poster hall here, but you'll notice there's much more space, so you can actually hear. Okay, and so let's say we're walking through the SFN poster hall, a little bit jet-lagged. I'm going to represent that as a koala holding a gigantic cup of coffee. And I walk through this poster hall, and I see tons and tons of amazing talks, posters, let's say. I have these great conversations with people. Some of them are on computational neuroscience, some of them with a clinical disorder, something like dementia, Parkinson's disease. I might see some really great work on human imaging, pushing the boundaries of cognitive function and mapping that in the human brain. And then I'll go and see a really awesome systems talk that's tracking calcium imaging, wide field calcium imaging during some complex task in a model organism. Now, the problem, I think, with these is that I've had a completely different talk with all four of these people. The languages that they use, the kinds of techniques that they use, their epistemology, the very questions they're trying to answer are completely different. But they're all studying in some way the same system, the brain. And I think if you think about it for a little while, I think neuroscience is actually a really young field. We're actually quite segregated and separated apart in a lot of the concepts that we use and the terms that we use and the ideas that we discuss. And I'd really, really, really deeply like to be helpful and a part of making that more integrated. So just to kind of further the toy example, let's imagine something like we took each of these different posters in these different groups that we saw, and we kind of compared the different concepts and terms, or maybe the different papers that they were citing. It's quite rare to actually come across papers that cite the same papers as the other fields. So if you're in a clinical field, it's rare that they'll be able to come to somewhere like Cosyne and read about the kinds of amazing advances that are being made. And vice versa, the kind of clinical problems that are really detailed. If you look at the abstract book, I'm sure lots of people in the audience think about disease a lot, but there's very few posters about disease. I think the word disease comes up 8 times in the abstract book. So it's one of these things that's, I think, a really big problem in neuroscience if we want to really integrate together. I will give you guys your kudos, though. Cosyne is actually doing an incredible job of connecting across these fields. And I know that this was the goal 20 years ago. It's actually a real honor to be here in the 20th anniversary of this amazing community. And I think you've done an incredible job linking together, and think of all of the benefits that have come from that. Think of all of the amazing computational questions that can now be asked in model organisms and animals that we just didn't have beforehand. And I think there's more benefits to be had if we can find the ways to link across. And let's imagine a problem, right? So I have a medical background. Imagine I'm a clinician sitting in my office. Someone comes in with a particular early onset dementia, and they've got a problem with certain cognitive issues. They can't really do things together in parallel. They can't multitask. If that happens, they just fall to pieces and everything goes away. I think it's really hard to actually link those different kinds of problems to the kinds of insights that we get in a model organism with the kinds of resolution we get in this meeting. It's really hard to train an animal to do one task, let alone multiple together. And you can definitely take approaches that we've seen these beautiful talks about compositionality and using RNNs to train these things to do these different kinds of tasks and push them together. But then the question becomes, is that exactly how the brain is doing it in an organism? And there's a lot of really fundamental questions. And what I'd like to offer today is not solutions, but maybe a little bit of an assist that I think human imaging is actually in a place where we can actually connect together and help. we can help draw some of these bridges between these different fields. So we do human imaging in my lab, but we also try to connect it with the kinds of concepts and ideas that are happening in this field with the hope that we can create bridges between the different areas. If you make those connections, obviously, we can build the network and we can make it more integrated. We can help to build that language together to get, I think, more satisfying answers to the kinds of questions we care about. When you think about this for a little while, you realize that there's actually a really big problem staring you in the face. The problem of scale. In a human imaging experiment, we might not know exactly what's in the signal that we've got. We might be measuring a big macro LFP or the bold response. But we can put someone into a context that's really, really subtle and nuanced. We can give them a particular cue, a language cue, and they can do a very subtle task, one trial. On the other hand, if you're looking down with the microscope, you're actually looking with exquisite detail, detail that we can only dream of in the human imaging field. And you can actually have causal manipulations in model organisms that we really just don't have access to for ethical reasons. So I think what we really want to do is find ways to connect across these different areas and get the insights from both to link together. So in my lab, we spend a lot of time thinking about neurobiology, reading the papers of this field, thinking carefully about neuroanatomy, but then also trying to think about what's being done in the neuroimaging space, the kinds of advances that are made there, the kinds of ways that we record human imaging. But we try to bind them together with computational modeling. That comes in lots of different forms. Sometimes we use spiking models, sometimes we use neural mass models, sometimes we use things like RNNs. Depends on the question at hand. But we find that the kind of emergent property that comes out of this kind of a challenge, it's not linear at all. It takes a long time. But if you can find those satisfying tethers between the different systems, different scales, you come up with really, really, really cool next step questions that I think you don't if you try to stay in one of the areas on their own. So that's what I'm going to try and convince you about today. So I'm going to try and throw at you three completely different concepts and show you how we can link them together via a particular area in the brain that I'm absolutely fascinated by, the thalamus. So this is the concept of different brain states, the idea that you can be awake or asleep, jet-lagged or alert, you could be anesthetized or conscious. The idea of perceptual content, the ability to track certain items in the world, or often perceptual illusions are the way that these are studied in the field. And this idea about combining these different skills together in these really interesting kind of dual task conditions where you're trying to do things at the same time, like drive home or have a conversation with your spouse. And so this is the area that I'm going to try and convince you is involved. And in first pass, I don't think anyone, if you asked them beforehand, would have said those three regions are related to the thalamus. Oftentimes you'll hear people describe things about a cortical region, or maybe they'll think of a particular part of the brainstem for arousal. But it's rare to think about this structure. And it's actually quite fascinating when you study it, because it's sitting in the middle of absolutely everything in the brain. It's highly conserved, bilateral structure in the diencephalon. It's connected deeply to the cerebral cortex, the basal ganglia, the hypothalamus, the cerebellum, superior colliculus, you name it. Right in the middle of it all. And so what I thought we could do today is just walk through some of the really interesting features of the thalamus and then try and see if we can link them to these different behaviors. All right, so here I'm showing you the thalamus again, this time in an axial slice of a human brain. So anteriors at the front, posteriors at the back. I've labeled the cerebral cortex and the basal ganglia for you enthusiasts. But here's our little thalami here. But even though they look like a couple little eggs sitting in here, they're actually not homogeneous. They have really, really different structures within them. So if you actually look down at the cytology and also at the different types of connectivity of these different parts of the thalamus, it's actually perhaps better to think of them as a collection of subnuclei. So I label a few of them here. There's the sort of ventral compartments that project out to the frontal lobe with the medial dorsal nucleus. The lateral geniculate, probably one of the most famous ones, receives inputs from the retina, projects them on into V1. The pulvinar is a really, really fascinating area that's expanded over recent evolutionary time and connects to the wide parts of the parietal, temporal, and occipital lobes. And this area here, the central lateral nuclei, are part of what we call the intralaminar nuclei, a really, really fascinating set of structures that are really deeply related to the arousal system. So now if we advance our understanding of this, we can think about the kinds of connectivity that the thalamus has with the cerebral cortex. And so it looks a little bit like a spaghetti diagram, right? There's all these different types of nuclei with different types of projections. A few things to kind of double click on is the diversity here, right? Rather than having that one type of projection from the LGN just to V1, there's many different types of connectivity in the thalamus. I know I'm showing you here in cartoon form, but you can track this down to the cellular level. And so there's some areas that have relatively diffuse connections, some that project really predominantly to the striatum as well as to the cerebral cortex across different lobes, and some of them have much more targeted projections. Now, if you go looking in the human imaging literature, as we did recently in a review paper, you find that the thalamus is actually associated with a wide variety of functions at the whole brain level. So if you went through and looked at human fMRI studies that have looked at the thalamus in different cognitive contexts, you can actually make really compelling arguments to support the notion that the thalamus supports activity in local areas, perhaps by boosting in a sort of divisive normalization-like computation the activity of one population at the behest of the others. It supports coupling between areas, let's say oscillatory synchrony in a gamma band during a working memory task between the frontal and parietal lobes, if we're talking about the pulvinar. It could support topological configuration. So here, this is now different patterns of connectivity, but separated from one another in the actual context of the whole brain. And it also supports the variability of local regions. If you inhibit the thalamus in a neurosurgical context, the areas that would drain to that thalamic nuclei become less variable compared to baseline. And so the question is, what is it that binds all of these things together? Here's this deep structure connected to the whole rest of the nervous system. It's associated with all these different functions. And I'm going to argue that it's going to be helpful if we zoom into the microscale. And once we're down there, we find a New Zealand anatomist. A simple rule for all of you that haven't visited Australia: if someone from New Zealand does anything impressive, we get to pretend like they're Australian. So for the sake of the talk, Ted Jones will be an Australian. So Ted Jones literally wrote the book on the thalamus, and he was really interested in trying to track the kinds of diversity that he saw there down when he looked at the cells. So the main way that he framed the kind of spread of different cells in the thalamus, rather than thinking about those little subnuclei, let's go down to the cellular level. It turns out that a really nice way to think about it is a spectrum between two different connectivity types. Now, in reality, there's actually a blend of these different types. Some of the cells have some of the features of both. But a good way to think about it is what we call the core nuclei, which have a really targeted projection, and the matrix nuclei, which have a really diffuse projection. Now, these are kind of interspersed in a little admixture in every single nucleus in the thalamus. The reason that they're really cool and that you can think about these as having a really different function is the interpretation comes from actually their projections to the cerebral cortex. So here what I've done is lay out a description of the cerebral cortex. So here we have the supragranular layers of the cortex, the granular layer here shown in the middle, and the infragranular layers. And the idea is that the core cells end up sending really, really targeted projections into layer 4, whereas the matrix cells actually send really diffuse projections to the supragranular layers, as well as this really interesting input to these cells here in layer 5A. So they have these really, really different types of topography, which I think betrays a potential different impact on the kinds of processing that can happen in the cerebral cortex. So if you think of Raj Rao's really lovely talk yesterday about the kinds of predictive processing algorithms that might be playing in the cortex, I think this will have a really interesting constraint on the types of messages that can be passed, the different types of ways that evidence could interact. So here's the core. If we contrast that with the matrix, completely different between these targeted projections and these diffuse projections. But sitting right in the middle of all of them is this really, really fascinating cell that we've used as a tether in a lot of our experimental work and our computational work. And that's this deep, thick-tufted, deep layer 5B pyramidal tract cell. So if we zoom in on this, some really, really beautiful work has been going on over the last few years that I like to broadcast every time I get a chance. From Matthew Larkham, he's also an Australian, he's sitting in the audience today as well, and Jan Aru, who's from Estonia. He's never been to Australia, I checked. And so Matthew has been studying these beautiful cells, these layer 5 pyramidal neurons, and to cut a very, very long research program short, one of the really, really interesting things about these cells is that they're basal compartments, they're, the axons that actually rise right into the cell bodies are actually really, really extensive and distributed away from the apical dendrites where they receive the feedback projections from different layers of the cerebral cortex. And the idea is that different kinds of inputs to these cells, and whether or not you have coincident input to the basal areas and the apical layers, can change the kind of firing output of the cell. Specifically, if an action potential happens in the basal dendrites, it sets off a calcium wave that rides up this apical dendrite, and then if it meets the inputs from the apical dendrites from other glutamatergic inputs, it can actually cause a nonlinear increase in the burst firing of that cell. Specific to our talk today, Matthew showed that the thalamus, the higher order thalamus, so those matrix-rich nuclei, that little projection I showed you that went into layer 5A through mGlu-R receptors actually gates the ability for these things, this system to couple together. So the thalamus is playing a really crucial role in changing the kinds of ways that the pyramidal cells, the main outputs of the cerebral cortex, can function. He's done some really beautiful work as well, showing that different types of anesthetics, so here ketamine and then 2 ether gases, actually decouple the compartments here in very specific contexts. But this gives us a really nice, interesting insight into one of the questions that we can ask, right? If we wanted to understand what it is about changes in brain state that are related to the thalamus, maybe we could have a look at this idea of the thalamus helping to couple these different compartments and see what happens when it breaks down. So Brandon Munn, a really talented postdoc in my lab, he's from Adelaide in Australia, did a really nice study. So he created a little spiking 2-compartment neural mass model, a neural spiking model. And he was interested in basically taking these 2 different compartments of spikes and having them combined together in different contexts. So he has a single parameter here, this beta parameter that's controlling the coupling between these 2 compartments. And so when the coupling is on, the cell can go from this somatic-driven spiking mode into this dendrosomatic coupled burst firing mode. He also wanted to control the spread of the interaction in this cell. Because those matrix cells that we mentioned have this interesting diffuse projection pattern, he wanted to know what happened if he tuned a different parameter as well that controlled whether there was a targeted projection or a more diffuse projection. And so he took these different cells, wired them up into a really simple Mexican hat array. He then coarse-grained their activity to make sure that we could mimic something like an LFP. And then he essentially went through and took 2 really interesting data sets. One's a macaque monkey that has natural sleep and then transitions to awake, which has really nice ECOG recordings across one of the hemispheres of its brain in the cortex. And then another is a human data set where people are given the anesthetic agent dexmedetomidine. And an interesting thing about dexmedetomidine, it's an alpha-2A agonist, which puts you into essentially a stupor, but you can still be roused. And then what they did is they roused them from the stupor, and then they asked them, what do you remember? And in certain cases, people just remembered being awake. In other times, they remembered being dreaming, and then other times they didn't remember anything at all. So what Brandon was able to do was to calculate a few metrics on these different data, and then plot them into this little 3-dimensional space. And then he used a swarm fitting algorithm, B-theme, to essentially fit these data to our parameter space. So now we have the monkey data here with the wake, the waking, and the sleep, and we have the human data with the wake, the dreaming, and the sleep. And what was really interesting to us is there was a convergence between some of the zones that were controlling the wake, particularly this area here, that was controlling the wake in this beta parameter. So the beta is about intermediate. Now you can do some bursting and some spiking, but you're doing it in a relatively diffuse way, the sigma parameter controlling that diffuse coupling. So we think that's a really nice evidence for this notion of the thalamus helping to control the kinds of dynamics we see during the wake state. But one of the things we really love about these models is you don't have to stop there. You can try and figure out why is it the case that the wake state is different from the sleep state in our model. And so here what I'm doing is I'm plotting now the activity of one of the trials in his model, and in the sleep stage and in the wake stage. So I've just picked a point in that parameter space that represented those 2, the locations of the swarm fit. And the green dots will tell you when there's a spike, and the orange dots will tell you there's a burst. So even though it's 9 o'clock in the morning on a Sunday, I'm sure all of you can tell that there's more of the orange on the bottom than the top, right? There's more bursting in the wake state than the sleep state. Not surprising. But the thing that was really, really informative to us is when we unpack this over time. So if you watch the top, you can see that these bursts will sometimes trigger another burst, often quite locally connected to that. The spikes will trigger another spike in these little chains of green. But in the bottom, what we saw is these really, really interesting bursting cascades, where essentially a burst-firing neuron would have a really increased likelihood of causing a burst in another neuron that was connected to it, and so forth. Now, the really cool thing about this is that although you're starting to be able to do this in empirical data in some model organisms, we could actually track exactly which of the neurons were connected to one another in our model. And we can actually show that you see, for the physicists in the audience, an avalanche effect. You see a power law of these different durations and sizes of these different avalanches. So there's a really, really interesting connection here between the thalamus and going across states and this microscopic mechanism and these kinds of features that we think might characterize the waking state. All right, so that's the micro scale zooming up to the macro. What if we started at the macro? So there was some really, really beautiful work that's just come out recently from some collaborators of ours in Wisconsin. You'll guess again, he's also Australian. He's from Melbourne, though. I'm from Sydney. Melbourne and Sydney, yeah, we don't really get along so well. But we collaborated in this project. Yuri's a really, really lovely experimental physiologist, so he works in macaque. And what they did in this really, really beautiful experiment is they gave the macaques propofol anesthesia. So this is a GABA-A agonist. And what they're going to do is they're going to anesthetize the animals. And they go into the central lateral thalamus, that red one I showed you before that projects to the striatum and out into the frontoparietal lobes. And they stimulated with 50 hertz. And what they were able to do is actually rouse these animals from anesthesia. And so here's a direct stimulation of the thalamus that wakes an animal up. This result was actually confirmed in 2 other groups independently as well. We used slightly different protocols, but the basic idea sticks, which is that if you've got an anesthetized animal, you stimulate its thalamus, you can wake it up from anesthesia. They also noticed that there was an increase in alpha coherence on the EEG they measured from the scalp. So Eli was really, Eli Muller in my lab, he's a Kiwi as well, so here he is staring longingly at Ted Jones, the other Kiwi. Don't worry, they're both Australians for the sake of this talk. Eli was really interested in whether or not he could model this effect, but he wanted to model it at the macro scale. Eli has a background in neural mass modeling and is really interested in this, the different kinds of intuitions that you can get from such an approach. And so what he did is he started with this really interesting quirk of evolutionary history, which is that both of these different cells in the thalamus deeply need to make sure they buffer calcium. Calcium's a big problem for them, but they use different types of genes to support the calcium-binding proteins for this. They both work basically in the same way, parvalbumin for the core cells and calbinin for the matrix cells. So then in a project we were able to use the Allen Human Brain Atlas. We used the expression of these 2 different gene types. We made a mask of the thalamus, and we were essentially able to get a time series, now in a resting state fMRI experiment, of the relative activity in the sort of bold activity sense of these different core cells and the calbinin cells. We could then regress them back against the time series of the rest of the cerebral cortex, and we could apportion to each of the different cortical parcels whether or not they were more connected to the core population or the matrix population. And we were actually quite excited and surprised to see, but in retrospect not surprised, that you can essentially find this really interesting dichotomy between sensory regions that are actually wired in, temporally coincident with the core population, so here in V1, S1, and A1, and then the matrix cells actually much more covariant with the posterior cingulate, the hippocampal formation, the medial prefrontal cortex. For those of you who don't read the human imaging literature, this area, or this set of areas gets a lot of attention as something called the default mode network, and people get very excited about it. We didn't want to overinterpret anything cognitively, though. We wanted to actually use this as a constraint for our model. So what Eli did is he built a neural mass model of the corticothalamic system that took advantage of this gradient. So essentially what he did is he took this little model here. So now rather than modeling specific cells, we're modeling different populations. And we've got a little core cell here and a reticular nucleus and an excitatory and inhibitory population in the cortex. And essentially they can whisk away in influencing one another over time. We can fit this to basic EEG data. And then Eli added this really interesting element of a diffuse coupling parameter. In some previous work, we've shown that actually just adding that diffuse coupling parameter changes the kind of activity that you see in the system in a really, really cool way. And for those of you who don't have backgrounds in physics, this was actually a really, really deeply helpful analogy for me, as someone who doesn't have a background in physics as well, to understand what might be going on under the hood. The idea is that as you increase the diffuse coupling to this system, what you're doing is you're sharing around activity. And that's a little bit like increasing the temperature of a glass of water. So that obviously increases the kinetic energy of the system. The water molecules start bouncing into one another. But a really interesting feature of when you heat up a water glass is that there's now a widening of the distribution of kinetic energies as well. And a way to think about that is that there's some water molecules in your little glass that just get bounced into by a few more water molecules than you might otherwise expect in a little window of time. They might think it's a little hotter than it is. And then there's another group on the other end of the distribution as well. So this is, we think, actually a really key feature of diffuse coupling in the nervous system that essentially allows you to have this really interesting spread of diversity of states. So in this model, Eli, let's return back to our corticothalamic model. Eli now has to give it an anesthesia, so he essentially mimicked propofol by upregulating the IPSP of the GABA-A receptors in the model. And then he went through and he stimulated specifically the matrix thalamic nuclei in the context of actually that gradient that we mentioned before that was biasing the connectivity of the system. And the challenge for us was, in the empirical data, If you look at this, the wake state, there's a particular level of alpha coherence in this particular frequency band on the EEG from the scalp. And if you give the animal propofol, the coherence drops. And Eli was able to recreate this finding with his model, which was awesome. But one of the things that we really like about this is that you don't just recreate that one finding, but many findings around the literature of people that have used whole brain imaging, things like EEG and fMRI, to differentiate the sleep and the waking or the anesthetized and the awake state. And we, again, think this is a really, really great feature of models like the neural mass model, because we can start to actually break down what's away from these different measures that we're using towards the process that might be supporting them. Again, we can also double click on these models and figure out what it is about them that makes them interesting, get an interpretation for why the matrix thalamus can actually help support the wake state. So what Eli did in this simulation is he stimulated 2 different regions of the cerebral cortex. So here I'm showing you a flat map. This here is the central sulcus, here's the front of the brain, here's V1 at the back. And he stimulated an area that was rich in the matrix connections in our resting state data, and an area that was rich in the parvalbumin core connections. And I think the really exciting thing is that there's a completely different character of traveling wave that emerges from that model. So if we plot this now as the time to peak here on the y-axis, with the ROIs sorted with their time to peak, you can see that in the case of the core stimulation, a neuron, a particular area in our model will get activated, and then it'll follow with this really interesting kind of linear wandering path down to the end till all of them have become activated. But in contrast, when we stimulate the matrix cells, we actually get this widespread activation, where the activation of one area in the model brings on the rest together in parallel. And so the really beautiful intuition behind this is if you stimulate a cortical area, they can then go down to the thalamus and then hit a target that can actually influence the rest of the network, or a large parts of the rest of the network. You've got this capacity to essentially prime the cortex to be ready for the next input in the sequence. So it's a little bit like this idea of bringing in the system up with a gain modulation that allows you to essentially process information in a completely different manner than you can in that serial processing mode. All right. All right, so that's brain state with 2 different perspectives, one starting at the micro, one starting at that macroscopic scale. What about perceptual content? So this is a little bit of a trickier one. So here again, we were inspired by Matthew's work. He'd done some really elegant experiments in the barrel cortex of mice, where they essentially trained a mouse to lick a spout whenever they felt the twitch of their whisker. And then they could essentially go through and whisk the whisker at different levels of intensity and plot out a psychometric curve. So it's really standard stuff in threshold detection tasks. And then what Matthew was able to do through a series of experiments, essentially take our little, layer 5B pyramidal neurons here. So the cell bodies that are in the deep layers, the apical dendrites that receive that feedback, and the matrix thalamus that's coupling their connection to force them to burst fire. And he could go through and he could experimentally manipulate different parts of this circuit. So he could provide an optogenetic activation of the apical dendrites, and he could shift this curve to the left. What that means is that the animal's now detecting more and more subtle whisker flicks than they were before you actually released the apical dendrites. You actually also have them have more false alarms as well. In other words, they detected a whisker twick even when it wasn't there. In contrast, if they put in the chemical baclofen, this is a GABA B agonist, which is going to prolong inhibition in the apical dendrites, you now shift the curve to the right. So now animals are not noticing when you're sitting there whisking their whisker at a position that they would have noticed before. And really importantly for the talk today, Matthew and colleagues in a follow-up study actually used a different approach to actually inhibit the targets of these layer 5 pyramidal neurons down in the matrix thalamus, as well as in the superior colliculus and the striatum. And they actually showed that if you blocked the subcortical projections of these layer 5 pyramidal neurons, you actually shifted the curve to the right as well. So this is a really interesting intuition. I think a lot of people in this audience might think that perception is just this cortical phenomena. If the area in the ventral temporal lobe lights up, aha, you've seen it. But here's some evidence that you actually need the subcortical system to prime it, to be involved in this circuit somehow. We don't know all the details, but I think it's a really interesting challenge for us to think about a more distributed explanation for these different types of phenomena. So a grad student in my lab, Christopher White, who couldn't be at the meeting, he's also an Aussie, was really interested in trying to model this. So here he used a model very similar to the one that Brandon had in his model, the dual compartment spiking model. he added in a little fast spiking interneuron to help control some of the activity in the system. But basically the circuit diagram is the one that I've been describing already today. You've got two compartments in these layer 5B neurons. They have this gating that is influenced by the matrix thalamus, and you have this sort of widespread diffuse coupling as well. We also do another interesting thing in this model, which is that we essentially we represent the ratio of different cortical to thalamic cells in the model as well. This mimics a feature of neurobiology that we've known about for a long time. There are essentially something like 100 to 1,000 more cortical neurons than there are thalamic neurons. So we have just a little simple 10-to-1 ratio here. And then what Chris can do is he can stimulate different parts of the cortex and then read out the burst firing in the model. And so here are those three different experimental results that I showed you before from Matthew's work. And in Chris's model, he can recapitulate all three of these. So I don't think this is, really that profound of a statement, right? This is a really simple model. It can only do a couple of different things. It can burst fire when there's coincident input. It's constrained by thalamic gating. But the fact that we can get things to go in the right direction is really exciting. But what Chris really wanted to do was to push this model into a different direction that's a little bit harder to study than in the lab. And that's to study a phenomenon known as binocular rivalry. So this is a mainstay of human research into visual perception, where the idea is that you see different stimuli presented into each eye. So let's say a red grating to the left and a green grating to the right. But rather than seeing some weird, mushy kind of brown cross in between your vision, what you actually end up perceiving is a red grating that then switches to a green and then to a red to a green in a really interesting kind of stochastic manner. If you're interested in this space, there's a really beautiful review by Srdjan Ostojic that you might like to double-click on, which goes into some of the computational ideas. And so what Chris wanted to do was essentially take this model that he created, not change a single parameter, and essentially force this model to do binocular rivalry. Now, it turns out for very simple reasons, it's actually quite hard to get mice to do binocular rivalry. They don't have as overlapping of a visual field, and so it's really hard to get the system to have this interesting binocular confusion. It is possible, and people are starting to do some really interesting work in this space. But Chris was interested in this challenge. And so without changing a single parameter, here's his model now conducting this experiment. And as he stimulates two different parts of this ring, creating a frustration or tension, he sees a switch that mimics the features of binocular rivalry. For those that are interested, he's able to recreate Levelt's laws, which is a kind of really crucial test of these kinds of models, and actually also recreate a number of different experimental effects that I haven't got time to go into today. But if you're interested, he's got a paper on bioRxiv that goes through all the details. Just for the sake of this talk, I also gave him a challenge. Can you go into the human imaging literature, find a prediction, and see if we can test it with your model? And so what he did is he found this really interesting prediction from a paper that's just been put on bioRxiv, where humans perform a visual rivalry task. They essentially, in this study, find that different parts of the pulvinar actually track the rivalry stimulus, the perceptually dominant stimulus. And when we take Chris's model, coarse grain it, and then put it through a hemodynamic balloon model, we can essentially recreate this exact same finding. So this is so exciting to me, right? As someone from the human imaging community, where we normally talk about a thresholded map that's some output of a statistical experiment, here now we can talk about a mechanism down at the microscopic scale about something we care deeply about, the thalamus and the cortical interactions, but we can map it up to the large scale and start to ask questions of human neuroimaging data. All right. I've noticed that I'm running a little bit late on time, so I'll go through this last bit a little quicker. So the last little challenge here is to go from states to contents to skills. What do I mean by that? This little kid here, it still marvels me. Look at this guy. Like, he's balancing on this little balance board with this little wheel thing underneath, little cylinder underneath it. He's got his little hockey stick doing his deeks. His eyes are like locked on some stimulus over here. He's got a fan going. He's doing this all in parallel. And I just find this a fascinating challenge. Like, how on earth is it that his brain is able to do this? I just think this kind of stuff is just so much fun to think about. And so, consistent with the theme of this talk, I want to try and convince you that the thalamus, again, is playing a really crucial role. But this time, because it can act as a way station or a kind of conductor of the subcortex. So it turns out that these different populations that I've mentioned today, the core cells and the matrix cells, actually receive completely different inputs from parts of the subcortex. So here, the wide field cells of the superficial superior colliculus project to matrix cells in the pulvinar. The globus pallidus internus actually tends to inhibit areas in the ventral anterior and medial dorsal nucleus that are matrix cells, which I think is actually inconsistent with some of the original ideas about how the corticobasal ganglia system was wired. And the deep cerebellar nuclei, although they're a little bit less selective than these other two populations, have a really, really strong driving glutamatergic influence on these core cells, particularly in the ventrolateral nucleus of the thalamus and the MD. So the question is, can we get some insights from some of these different systems and how they're organized and figure out whether or not that might help us understand how this kid performs this incredibly complex skill? Why do I think this is important? Well, in cartoon form, the topology of these different circuits could not be any different. Right? In the basal ganglia, we have this massive collapse of dimensionality from billions and billions of cells down to very, very small populations, which then fan back out in the matrix population. The colliculus also has this really fascinating collapse and expand architecture. But the cerebellum does something kind of different. It does a collapse, then a big expand, then a collapse, and then a really interesting driving function. And I think if we start to take these kinds of topologies seriously, we're going to get really interesting insights into the kinds of computations that can emerge from these kinds of circuits. So the cerebellum is a really, really fascinating structure. There's not really a great way to kind of pour a sort of a lasso around its functions. Just thought I'd double click on a few of the different intuitions that we can use for what it might be doing. There's some really beautiful work showing that the cerebellum is really ideally organized as an adaptive filter, a little bit like noise canceling headphones. There's some lovely work by people in this field, Kaiko Gajic and Litman Kumar, showing that essentially the inputs and the spread to the granule cell population are actually a really great way to get a linear classification out of a massively overlapping input. And there's also some really lovely work by Michael Mork's group in Texas showing that you can actually condition on the outputs of the cerebellum, and you can essentially train an animal to take an unconditioned stimulus, pair it with a conditioned stimulus, but also pair multiple different responses in time. So this interesting idea, how do we combine noise-canceling headphones, a splitter, and a loop pedal? Like, what is that, right? I don't know. But I think I would like to argue today that this kid's using something like this, right? Every time he moves his leg to the left, he's starting to cancel out the sensory consequences of his action so that he can focus on the particular variable that he needs to do to balance himself on this board. He's splitting the different information into different streams. He doesn't want the deke that he's performing with his hockey stick to influence the balance that he's doing. And he's doing this loop pedal type function as well, because the movements that he has with his stick, he's not focusing on every little detail. They're flowing out in a sequence. So I think if we can start to understand some of this topological architecture of the cerebellum and how it interacts with the thalamus and the cerebral cortex, we'll be a step further towards understanding and having better explanations for this kind of behavior. A step in that direction, Eli, again, did a really, really fun study. We took an open data set from a German group where they had someone perform a task that I would challenge you would be very, very difficult to perform in a bee, which is to have them on a balance board where they're controlling an avatar that is balancing back and forth. They have to move it. And then in a separate trial, they perform a calculation task, serial sevens, and then they perform them together. And our prediction is that this harder dual task condition, where you're having to both calculate and balance at the same time, should recruit a widespread corticocerebellar network. And lo and behold, we find really lovely evidence for that. There's a bunch more in the paper that I don't have time to click on now. But I really think this is a super fascinating area to start to think about these really complex cognitive concepts and how they might relate to neurobiology. I also unpack a lot of this stuff in way more detail if you're interested in a perspective paper where we kind of think about the topology of this different circuit and try to map it into the language of attractor landscapes and low-dimensional trajectories, thinking about the idea of the cerebellar nuclei being able to drive a channel in an attractor landscape. And I think there's some really, really interesting discussions we can have as a community. All right, so wrapping up, I've tried to convince you today that these kind of three completely different concepts, states, perceptual contents, and skills are all related to the thalamus in this area that we really don't focus on, I think, near enough. But more importantly, maybe the kinds of ways we've been thinking about the nervous system are kind of flipped around, right? We often have explanations of higher complex behavior that are all in this little circle here in the cerebral cortex. But the thalamus has this really, really interesting position where it can interact with the cortex and then gate the basal ganglia and the cerebellum's influence as well. If you're interested in hearing more about this kind of stuff, I'll be talking in the workshops about another absolutely favorite area of mine, the ascending arousal system, which performs this gain control modulation over this entire circuit, and I think has a really, really fascinating role to play. So that's on Tuesday. But more broadly, I wanted to come back, let's zoom all the way back out and say, here I am, I'm coming from a different community. You have to understand how deeply intimidating it is to speak to a room of such beautifully intelligent people who are working on really incredible science. It's the science that I aspire to do, and I want to be really clear about how admirational it is to be in this field. I've tried to convince you that we can use neuroanatomical detail and whole brain imaging, and we can link them together with biophysical modeling in ways that provide new, exciting hypotheses. But that's not all we can do. And if we want to take advantage of this opportunity in front of us to take the human imaging community and bind them together with this field, there's lots of other stuff you can do. The one that we really need a lot of help with is figuring out what on earth the bold signal is. It would be really, really useful for us if we can make specific predictions. If we see bold go up, what kind of cells should we see down below? And it might be an underspecified problem, but let's figure that out together and see if we can do better science. And so with that, I'd like to say thank you so much and really highlight the work of Eli, Brandon, and Chris. Thank you.
Okay, thanks, Mac. We have only 1 minute for questions. If you have a question, come on up to one of the mics and ask it. And if the next speaker can get set up while we're taking a question, that would be great. Okay, I'm not. Oh, one in the back. Yes.
Hi. Hi. Yeah?
Oh.
Yeah, thanks. Beautiful talk, actually. I share a lot of thoughts that you mentioned. Just because you mentioned it, do you have an intuition between the connection of the bold signal and, let's call it, the spike? Because I have the same question. I have no idea what the bold signal is. Thank you.
Yeah. I'm here asking for your help to figure it out, I think. Yeah, look, so there's some really beautiful work that was done in anesthetized monkeys by Nick Logothides' group, where they essentially could go in and have deep electrophysiological recordings of these monkeys during an anesthetized state. And they found a really strong correspondence with the local field potential. Now, what makes a local field potential is another really great question that I think we still need to figure out. In future work, other groups have done really elegant work where they drive different pyramidal neurons with optogenetic control and then measure the bold response and find really direct correspondence between pyramidal neuron firing and the bold response. But I don't know how selective it will be. We're really interested in understanding this better. And the other space that I think is really cool is people doing wide-field calcium and bold at the same time. So Todd Constable at Yale is the person in my community that I know that was doing some of the work, but I know a lot of people here were part of that. Essentially trying to find out which calcium transients were related to which bold signals and vice versa. And I think if you ramped that up and looked at different Cree lines of different cell populations, we could get a really, really good logic for how this stuff works together. But huge problem that needs to be solved.
Thanks, Mac. Sorry, we're going to have to move on to the next speaker. Our next speaker is Othman Lahrach. He's a PhD candidate at École Normale Supérieure in Paris. Othman, take it away.
Hello, Cosyne. Thank you so much for the invitation. I am very happy to present my work today entitled Comparing the Geometry of Static and Dynamic Coding Across the Auditory Pathway. This work is a collaboration with the Bathellier Lab, Sophie Bagure and Brice Bathellier, that provided us with the dataset, and my supervisor, Srdjan Ostojic. What is neural geometry? In general, when we try to understand how the brain represents sensory information, many attempts have been made at characterizing the tuning of individual neurons, such as the frequency and amplitude tuning. Another powerful approach is to study how a sensory stimulus is encoded in the entire population of neurons. To that end, we can represent the response to the stimulus in a space where each axis represents the activity of one neuron. In this case, I have represented the response to the sound here as the point S1 in the neural response space. In order to study geometry, we need many more points. If we have many more stimuli, we can plot their coordinates in this neural response space. And it starts forming a certain structure. The responses are not randomly distributed in this space. And this is because sensory stimuli are organized with a certain logic. Physically, they have a frequency and an amplitude, and also neurons are connected together and therefore correlated. So neural activity doesn't take up the whole space, but is rather organized. One way to study auditory response geometry is to have a whole range of sounds that cover the entire frequency and amplitude space. Here, we have used 33 tonic sounds covering 11 frequencies from 4 to 37 kilohertz and 3 different decibel levels. Sounds that are similar in frequency and amplitude are expected to have neighboring responses in the neural activity space. Therefore, we expect that the responses to this entire set of 33 static sounds would form a smooth surface. We will try to verify the veracity of this hypothesis and attempt to characterize the geometry of the manifold, which is the surface that we obtain. In vivo, Sophie Bagure and Brice Bathellier provided us with a rich dataset throughout the auditory hierarchy, spanning the inferior colliculus and the thalamus, with hundreds of neurons recorded in electrophysiology, and the auditory cortex, with 1,000 neurons units that were recorded in 2 photons. Crucially, the mice are awake, head-fixed, and passively listening. The sounds are constituted of 500 milliseconds of silence, followed by 500 milliseconds of sound presentation, and 1 second of silence again. The most important sounds that we will use are the static sounds from my previous slides, 11 different frequencies and 3 decibel values. We also have sounds that change through time, that either ramp up, increasing in amplitude, or ramp down, decreasing in amplitude. All of these sounds are repeated 15 times in random orders. This is an example trial of the data that we work with. We have neurons and time bins, and we have repetitions and different stimuli, so it's many dimensions. To study response geometry, neuroscientists often time average the responses. It's often done without assuming much, but it implies that we expect the neural code to remain stable in time, meaning that any change in the neural response is due to changes in stimulus properties. Let's try this approach first, before comparing it to what we obtain when we also consider the temporal dynamics. We will start by time averaging the responses. Let's visualize the inferior colliculus manifold here. Each dot represents the response to a given repetition of a sound presentation. The color represents the frequency, and the size of the dot represents the decibel level, or the amplitude of the sound. The continuous lines correspond to predictions of a Gaussian tuning curve model, a static coding model, that only depend on frequency and amplitude. As you see, the model reveals that the manifold is structured with 3 arches stacked on top of each other. Each arc corresponds to a given decibel level, and each arc displays a continuous frequency tuning here, shown as the color gradient. Remember, only 2 latent variables predict a sizable part of the variance in this very high dimensional space. We have R squared of about 0.3 to 0.6. This structure holds true for the 3 areas that we observe. 3 stacked arches with each arc corresponding to a given decibel level, and each arc displaying a continuous frequency representation. So far, we have studied the geometry in a time-averaged way. However, neural responses to sound exhibit striking dynamics, even when stimuli are constant in time, like here. So does studying the geometry of the manifold with a dynamic approach yield any different results? Okay. Each session is constituted of 3 main temporal segments. An onset at the very beginning of the sound, here in green, a sustained response in blue here in the middle of the sound, and an offset after the end of the sound, shown here in orange. In order to determine the subspaces at each of these key stages, we take 100 millisecond bins of the data within each of these periods and perform PCA. We keep the top 3 components to determine the onset space in green, the sustained space in blue, and the offset space in orange. Within these spaces, we will later on project the response manifold through the entire temporal sequence. To simplify, I will only show you a projection of the 80 decibel arc of the manifold in the inferior colliculus here. On the first line, we see its projection in the onset space here. Across time, we notice that this projection remains stable in time, implying that the inferior colliculus responses remain aligned with the components that were already present at the onset. If we do PCA at the middle of sound presentation, here, the projections are also stable in time, meaning that the onset and the sustained responses exist within a similar subspace. And finally, when we do PCA at the offset, it reveals components that are also aligned with the sustained response and the onset response. So the activity spaces seem to be very stable in time in the inferior colliculus. In the auditory cortex, however, this situation is very different. The onset space only contains patterns that align with the responses at the beginning of the sound. Then the responses fade out from this onset space. If we do PCA in the middle of sound presentation, it reveals new components of activity that were absent at the onset and that disappear after the end of the sound. Finally, if we do PCA after the end of sound presentation, we identify a completely novel space, here in orange, that is orthogonal to the previous patterns of activity. Let's visualize it better. In the inferior colliculus, you will see the manifold appearing simultaneously in all three spaces. And at the end of the sound, it will fade out simultaneously. In the cortex, the manifold first appears in the onset space before fading out while growing in the sustained space. And finally, it disappears from the sustained space and appears in the offset space. So there is a well-orchestrated subspace transition in the cortex. To summarize these results, here are the cross-temporal PCA variance explained matrices. So we notice a blocky structure in the inferior colliculus indicating stable activity spaces. In the auditory cortex, we see a clear signature of subspace transition at the level of the offset. And there is even a strong suggestion that there is a similar subspace transition at the beginning of the sound between the onset and the sustained period. In the thalamus, it's a bit of a mixed bag. So far, we have only static sounds. So in the absence of changes in the stimulus, could these subspace transitions be explained by recurrent connectivity? To test that, we designed an RNN where each unit is paired with a real cortical neuron. We initialize the RNN with the state of the cortex at a given time, 0. And we train it to predict the next H steps using the stimulus properties for these time steps. Using the predicted responses of the trained RNN, we can perform similar cross-temporal PCA analysis. So on the right, you see that the RNN displays similar signatures of subspace transitions that we had observed in the cortex, and these were absent from the inferior colliculus. What are the implications of these strong cortical dynamics that we observe? On the left here, I am showing you the static coding scheme, where responses have no time dependence and are only determined by the properties of the sounds. If we take a pair of symmetrical sounds, let's say one that ramps up in amplitude and one that ramps down, the predicted responses would also be symmetrical, meaning if we time average them, we should not be able to decode whether the sounds ramp up or down. This is a prediction of the static coding model. However, we showed that the auditory cortex deviates from the static coding scheme and displays striking temporal dynamics. How does that affect the predictions of the static model? On the right, I show you figures from a test in the 3 recorded areas. We time average responses to pairs of up and down modulated sounds, and we try to find an up-down axis. This is trials cross-validated and spans different values of frequency, so it's invariant to frequency. In both the colliculus and the thalamus, time average responses to up and down ramps seem undistinguishable, consistent with the predictions of the static coding scheme. In the cortex, however, we can easily decode ramp direction from time averages, inconsistent with the static model. Therefore, we propose that cortical dynamics, and particularly subspace transitions, could be a major contributor to breaking temporal symmetry and allowing us to distinguish ramp direction in this example. To give you a better intuition of this mechanism, let's use an analogy. Whenever you hear a sound, your brain throws a party. And just like a party, auditory responses are structured with a before party, the onset, a main party, the sustained period, and an after party, the offset. In the inferior colliculus, the party is quite basic, static and boring. It stays in the same space, the living room. In the next morning, an external observer that looks at the damage done to the living room would be hard-pressed to tell if the party ramped up or down in intensity. In the cortex, just like at Cosyne, the parties are more dynamic. They start in the kitchen, then move on to the living room and finish in the laundry room. Now, in the next morning, if an external observer, let's call him Tim, finds a pristine kitchen, a dirty living room, and a trashed laundry room with a broken washing machine, he would know for sure that the party went crescendo. This is how cortical transitions organized in time can allow us to disambiguate the order of a sequence of events only by looking at the average outcome. To summarize, we found that 2 latent variables, frequency and amplitude, are reflected in the time average response manifolds. Activity spaces remain stable in the inferior colliculus, which corresponds to a static coding scheme, but show striking dynamics in the auditory cortex. These subspace transitions can be reproduced by a recurrent network model. Surprisingly, the static time average manifold encodes sound modulation direction. However, this encoding axis that we observed in this time average scheme could in reality emerge from dynamic activity subspaces. Thank you for your attention.
Okay, questions? We have a few minutes.
Yeah, thanks. Thanks for the talk. So I was just wondering, I'm sticking with your party analogy at the very end. Is it so much that the rooms are changing or the participants are changing? So can we just look at which neurons are firing in auditory cortex and see that they're different in different phases, whereas in IC, there's actually the same neurons in every phase?
So you're asking whether it's a different population of neurons driving these changes. So we see mixed temporal selectivity in the neurons in the auditory cortex, indicating that at least a significant part of these neurons is participating to several stages of the party.
I see.
It's not distinct groups.
Thanks.
Hi, really cool work. I was curious, oh, it sounds like it's on now. Okay. I was curious, given that there's so many projections from auditory cortex back to thalamus and IC and connections within these areas, how does that impact the representations you're seeing in each area? And do you think it would change with more like contextually relevant sounds, for example?
Yeah, very good question. So we made sure to record the central part of the inferior colliculus with electrophysiology to make sure that it's not the part that is known to receive the feedback. We have also 2 photon recordings of the dorsal part of the inferior colliculus, and the results are a bit different from this, but they are too noisy to show here at this presentation. So indeed, the feedback seems to change a bit the representation and the activity in this, at least in the inferior colliculus, between the central part and the dorsal part. In the thalamus, there is also some strange phenomenon with respect to the code that seems to switch or to have like an additional axis that appears in the middle of the sound presentation. But this effect is pretty late. I think it arrives between 200 and 300 milliseconds. So I don't know how to interpret it in terms of feedback from the cortex. Yeah, but it's a very good question.
Okay, we can take one more while the next speaker gets set up.
How much of this phenomena can be explained by onset, offset, and tonic single-cell properties in the different regions differently distributed?
Yeah, so I think it cannot be dissociated from the single-cell phenomena that we observe. I mean, I think there are 2 competing but also complementary views where the single-cell phenomena would be driving the entire population or the result that we see at the level of the population. Some might argue that it's really this deeper computation happening at the level of the entire population that are observable as this onset and offset responses that we see. So it's really hard to give a clear-cut answer to this question, but it's very important to always try to relate these 2 levels of description. Well, thanks a lot.
Thank you. Okay, our next speaker is Ayu Lo, and Ayu is a research analyst at the Flatiron Institute in New York.
Hi, everyone. My name is Ayu. I'm a research assistant in Professor SueYeon Chung's lab at the Flatiron Institute and NYU. And today I'm here to talk to you about the geometry of hierarchical category structures in the monkey IT. So the motivation for this project is that humans have a tendency to organize knowledge in order to store and share information efficiently. A number of psychological and behavioral experiments have suggested that humans categorize visual objects hierarchically based on abstractions of common and shared attributes. From past work, we also have no evidence that suggests hierarchical categorization in the brain. In 2007, Kiani et al. Gathered a set of images of animal faces, animal bodies, human faces, and other objects, and they showed these images to 2 passively fixated monkeys. And then they made single-cell recordings in their anterior IT cortex, which is the final stage in the visual processing step. They perform dimension reduction on these neuroactivities, and they found that, the activities elicited from animate objects tends to be closer together, while activities elicited by inanimate objects tends to be clustered together as well. Furthermore, they also found that within these animate objects, you find cluster of activities from face categories, hand category, and body category. And a further breakdown within the smaller cluster, you can also find finer clustering of activities from monkey phase and human phase within the phase cluster, for example, that is still distinguishable. They then use clustering algorithms on the neuroactivities and discern the following hierarchical tree for the object categories. And as it turns out, the neuroactivities actually construct an intuitive category structure. Similar structure here is observed in the human visual pathway when the same stimuli set is presented to human subjects. So these past works are telling us that there is some hierarchy structure in the neuroactivities, but they rely on dimension reduction techniques. And they do not really tell us anything about the specific structure in the full neural state space. To study this question, we analyzed the population neural representation at different levels of the hierarchy. We have 3 different hierarchical levels in this tree that was discovered in the original paper, with the bottom being referred to as subordinate categories. And in this level, we have examples of monkey face, human bodies, animal bodies, and cars. Now, combining these subordinate categories and following the tree structure, we would get entry-level categories, such as the face, the body, and the object category. And finally, the tree would combine these entry-level categories into the superordinate categories, animate and inanimate objects. Grouping the population response to a given category at a level of the hierarchy, we can create different sets of neural population representations that we can study. We define each of these neural populations as neural manifold, these little structures here, where a point on these manifolds would indicate a response to a trial at which the image to a specific category was shown. So for example, to get this human body manifold, we would gather all the population, like some population responses to images where the human body was shown. And then to go up the level and get a body manifold, we would gather activities of responses to images where either animal body or animal body was shown. And finally, at the animate manifold, you would just get the responses from face, human body, animal body, hands, etc. So now that we have manifolds for each level of the hierarchy, we want to understand the structure and the organization of these manifolds and how they could change across the levels of the hierarchy. To do so, we employ the manifold capacity theory that was developed by Chung et al. In 2018 and 2020 that categorizes geometry in the full neural state space. 2 such properties are the manifold center distance and the manifold size. The manifold center distance basically describes how close together your manifolds are, while your manifold size describes how large the manifolds are in that neural state space. The theory also introduces a measure called capacity that quantifies representational efficiency, or how many manifolds the population of neurons can linearly separate. Capacity, as it turns out, is actually shaped by geometric properties of center distance and center manifold size. As you can imagine, as your manifold distance, center distances get larger, your manifolds are further apart, and separability is easier, and that means that your capacity is bigger. Conversely, if your manifold size gets smaller, then separability is easier, which also corresponds to larger capacity. So given this framework, we have a tool set to study the structure of our neural manifolds at each level of the hierarchy. We can talk about how far apart your manifolds are, how large they are, and how efficient they are for linear separation. So to begin applying this capacity theory for each hierarchical level, we also want a control set of manifolds to ensure that the clusters from the original paper give some meaningful category structure. As in, it is meaningful to put like human faces with monkey faces instead of human faces with human bodies, for example. As I mentioned, we form neural manifolds at each level of the hierarchy tree discovered by the original paper, and then we measure the capacity and geometry at each level. In our analysis, we also ensure that all manifolds share the same sample size so that the change in geometry is not due to the varying sample sizes. This limits our manifold to each composed of 15 samples with 319 dimensions. Then to build our control set, we shuffle the category labels, here indicated by the color frames. We shuffle the labels at the image level and then create like a random hierarchy from the shuffled labels. The expectation is that if there is category structure, then the manifolds under the original label should have a better capacity than the random labelings, meaning that the original labels are the better choice of grouping than their activities. So is there category structure when we compare original label manifolds with the randomly labeled? The answer is yes. We observe categorical structure at the subordinate level, as the capacity for manifolds under the correct labeling is larger than under the random labelings. We also see this across the other levels of the hierarchy as well. We also observe that there is a change in capacity across levels of the hierarchy. Specifically, there is a drop in capacity in the upper levels of the hierarchy. We asked then what could possibly be causing this change. Well, here's a hypothesis. In the original paper, neural activities for the subordinate categories are clusters of point cloud that then form larger clusters for entry and superordinate categories. Assuming that this is the same scenario in the full neural state space, this picture would explain the drop in capacity because the upper levels manifold are clearly getting larger. Right? Here are your subordinate levels, and then following your entry, and then this is being your superordinate manifold. Clearly, the size is getting bigger. And also, they are appearing to be getting closer to each other, meaning that the center distance is also decreasing. And according to our capacity theory, these two phenomena would lead to a drop in capacity. So since we observed this decrease in capacity in our neural data set, we are expecting that this would be happening in the neural data as well, that that the average center size is increasing and the average center distance is decreasing across levels. And let's see what really happens in the neural data. Well, surprisingly, the manifold size changes little across levels of the hierarchy. But while this happens, we do see a decrease in the center distance, which suggests that it is the manifold's location that is primarily driving the change in capacity. And furthermore, this would mean that our first hypothesis is incorrect. And the full dimensional structures of the neural population response is actually different from the lower dimensional observations that was observed in the original paper. So since the manifold width doesn't increase in our neural data, it brings up a question for what kind of manifold organization would prevent size from increasing. As part of our ongoing work, we offer two possibilities for this to occur. In one case, the manifolds are low dimensional and highly correlated along their axes. To picture the scenario, imagine, for example, papers being stacked on top of each other. Imagine that the papers are so thin, or in other words, very low dimensional, that the union would make very low changes in the overall size. In the second case, the manifolds are highly dimensional with low correlation along their axes. The manifolds with such high dimensional end up having very many intersecting subspaces, so that their unions also do not affect manifold size. We can distinguish between these two hypotheses by extending the capacity theory to include other properties of our neural manifolds, such as axis correlation. There are also things such as center correlation and center axis correlation. And this extension has, in fact, been recently developed in our lab through the works of Marklu et al. And Cho et al. There's also a poster on this recent work in this conference. On the other hand, we could use some creative analyses. Unfortunately, I don't have time to go into details, but the analysis points towards hypothesis 3 being the potential winner. So to conclude, we use capacity theory to discover category structure at each level of the hierarchy from the original paper. The theory shows that center distance is primarily driving the change in capacity over levels of the hierarchy. And with minimal change in the manifold size that we observe in neural data, we are observing an organization in the neural state space where other elements, such as correlation, might be coming into play. So going forward, we can use the new capacity theory with measures of manifold axes and center correlation. And also, I also want to point out that we do see the structure in hypothesis 1, but in deep nets. And that suggests that, to us, there is room for improvements in using the deep nets as models to the visual pathways. So I just want to end my presentation with acknowledging my collaborator, Professor Roozbeh Kiani and Professor SueYeon Chung. I'll thank everybody in my lab who gave me great advice on this project, with special shout out to Yuelin and Albert.
Okay, thank you. We have a limited time for maybe one question. If the next speaker could come and get set up while she's answering it. If nobody's making it to the mic just yet, I'll ask you a quick question.
Yeah, I have a question.
Oh, go ahead.
I'm curious, like the previous talk we just saw about how these manifolds can change in time in response to like an auditory stimulus. I'm curious how time could be incorporated into your capacity theories and like how that might impact calculating how many of these manifolds can fit together in a space.
I'm probably not the best person to ask about this. That's probably a better question for my collaborator, but I think it is possible to create manifolds based on the time trials, and I think there is some work going on in the lab that is working on that.
Cool, thank you.
Okay, thank you. Okay, our last speaker in this session is Yuelin Shi. Yuelin is a PhD candidate at Caltech in California.
So hi everyone, my name is Yuelin. I'm a graduate student in Doris Tsao's lab at UC Berkeley/Caltech. Today I'll be talking about rapid concerted switching of the neural code in inferotemporal cortex, another IT cortex talk. So I think this project strongly challenges the current way of thinking how object recognition works. For a long time, people thought object recognition relies on specialized modules in the brain dedicated to specific categories. A prominent example is the face processing network in primate IT cortex, including human FFA area, as well as the macaque face patch system. Many psychological and lesion studies suggest that face processing relies on specialized mechanisms dedicated to process upright faces. For example, we're much better at discriminating face parts presented in the context of a whole upright face compared to an inverted one. This leads to the suggestion that a face detection gate is implemented prior to specialized face feature processing. However, with the advent of deep neural networks, especially the introduction of AlexNet in 2012, a new view starts to challenge this old idea. The units in the deep network process all objects the same way, agnostic to the category of the input. Yet, one can find category-selective units in AlexNet, including face-selective units, gadget-selective units, and so on. But each of these units is applying the exact same sequence of filters to all images. If we view IT cortex as a deep network, then face patches may be formed simply by anatomically clustering all the neurons that happen to prefer the same category, while still using an entirely general mechanism for image encoding. Indeed, it has been found in IT cortex that neurons in IT can be well modeled by AlexNet FC6 units. And this similarity structure of Alex-style representation of objects can even predict the anatomical organization of IT cortex into 4 distinct subnetworks. So we set out to study this long-standing debate of whether IT cortex uses specialized coding mechanisms involving dedicated code to specific categories, or a general-purpose coding mechanism for object recognition. If there's a general-purpose code, then we should be able to use the decoding model learned by owning training with non-face objects to reconstruct faces that the monkey sees. And this will be impossible if the code for faces is a specialized one. So how exactly are we going to test the idea? As a first step, we need to parameterize all the objects. For every object, we can pass it through a deep network to get out a few thousand numbers describing this object at a high level, which can be used to classify the object identity with a linear classifier. And then we can use principal component analysis to further reduce the dimensionality of this space to just 60 numbers. Now, each object can be uniquely described by a 60-dimensional vector. And then we can record responses of a face cell to a bunch of different faces. We can model the responses of the cell as a linear combination of the 60 features of each face. Similarly, we can measure the responses of the same cell to a large set of non-face objects and get a set of 60 coefficients that model its responses to non-face objects. If scenario 2 is correct, then the 2 models we get should be exactly the same. On the other hand, if scenario 1 is correct, then there's no requirement for these 2 models to be the same. Indeed, it's even unclear whether the cells would have a well-defined, robust model in response to non-face objects. We call these coefficients the preferred axis of a cell, because a simple geometric interpretation of linear regression is that cells are projecting incoming features onto an axis. Our lab has indeed shown in an earlier paper that a face cell responds the same to all faces in the plane orthogonal to its preferred axis. Here, for our purpose, we call the preferred axis mapped with faces the face axis, and the axis mapped with non-face objects, the non-object axis of the cell. Now the question becomes whether the face axis and the object axis of a cell are identical. Again, if IT cortex uses a general purpose code, then there should be only one single axis for a unit. So how do these 2 axes compare for actual face cells? Let me first guide you through one example cell. First, let's plot all the objects that we show to the monkey in the feature space. Here, each dot represents the position of one non-face object in the feature space. For visualization, I'm only showing the first 2 dimensions of the feature space, which is actually 60-dimensional. Non-face objects mostly reside in this bottom left area. Here, these dots show the positions of the face stimuli in the same feature space. Faces all cluster in the upper right quadrant. Their features share a lot of similarity, and also they cluster separately from the non-face objects. Now, we can color all the non-face object dots based on the responses of the example cell to each of these images. We see a response gradient in this direction, which suggests that we can indeed use a linear axis to describe the cell's responses. We calculate the cell's object axis, and its first 2 dimensions are shown as the green arrow here. Now, what happened to faces? We can again color all the face dots based on the cell's responses to these face images. Similarly, we can get the cell's face axis using the features of the faces and the cell's responses to these faces. Strikingly, the cell's face axis and object axis are totally different. In fact, they almost point to opposite directions. So this cell uses a different encoding model for faces compared to objects. Here are object and face axes computed for all the cells that we recorded. The object axis all pointed consistently to this upper right direction, which is where faces are located in the feature space, whereas face axes are much more scattered. This result shows that face cells in general are using 2 different axes for faces and objects. But how can a cell decide which axis to use immediately when it encounters an object? One possible solution is that the cell is changing its encoding axis in a dynamical way. Here, I'm showing the response time course of the same example cell to all the faces that we show and all the objects on the bottom. For calculating the axis so far, we have been using response averaged across a relatively long time window. So for the next set of analysis, we decided to use a short 20 millisecond sliding window, so we can see in detail how the axis develop across time. Now, I'm about to show you a few snapshots of the cell's axis at different time points. Here are the face and object axis for 40 to 60 milliseconds after stimulus onset. This is before any significant firing of the cell starts, so note the axes are not so meaningful here yet. 80 to 100 milliseconds covers the initial peak of the cell's responses. Surprisingly, the face and object axes at this moment are actually pointing to the same direction. Then, after another 20 milliseconds, something more dramatic happens. The object axis stays at roughly the same direction, but the face axis rotates away suddenly, and this rotation is maintained at later time points. We found that the initial alignment and later rotate away of face axis happened to most of the face cells, suggesting there is a concerted change across the population. So far, I've shown that the face axis flips relative to the object axis in the first 2 dimensions. What about the higher dimensions? Well, the higher dimensions also reverse their directions. Here is a histogram of the correlation between the face axis at early and late time windows for low dimensions. And here is the same plot for high dimensions. In low dimensions, we see anti-correlation. In high dimensions, we see uncorrelation. So the face axis is changing in every dimension between early and late time windows. And here are the same plots for the object axis. The axes are positively correlated across all dimensions. So the object axis is not changing over time. To better quantify this temporal change, we plotted the similarity matrices comparing axes between different time points. Averaging across the whole population, the cell's object axis stabilized after 50 milliseconds. Similarly, the cell's face axis starts to stabilize after 120 milliseconds or so. So what about the correlation between face and object axis? Just like the one cell I showed you before, the cell's face axis aligns with their object axis in the beginning and then becomes negatively correlated with them over time. So to recap here, we found that a cell's face axis aligns with its object axis in the beginning, meaning that it's using a general coding mechanism. Then face axis diverges from this general axis, and this divergence only occurs for faces. So face cells achieve domain specificity over time through a rapid concerted change in the neural code. To our knowledge, not only has this code change phenomenon never been reported before, but even the idea that domain specificity could emerge over time has never been suggested as a solution to the debate of whether IT cortex is specialized or general. This tells us that IT cortex is definitely not just a feed-forward DNN, but must be using recurrent computations to produce the dynamics that we see. And since these dynamics occur each and every time in response to perfectly clear faces, we believe recurrent computations are part of core object recognition. So lastly, what's the function of this code change? When the face axes initially align with their object axis, they all point towards this face quadrant. We think this early phase is for face detection because most of the faces, regardless of their identities, will trigger the cell to fire strongly. For the long latency, we speculated that the code change is to improve face discrimination because the reversal in low dimensions reduces the initial high firing rate to faces, so that the cells now can use more bandwidth to represent fine differences in face features. To further confirm this idea, we tried to reconstruct the faces that the monkey see using either the early or late epochs of neural activity and see how much information can be restored from each. We leveraged a variational autoencoder to reconstruct faces from the neural responses to original images. Here is the reconstruction results of one example face. I'm showing the original face on the left, we found that the faces reconstructed using the long time window is much closer to the original image than using the short latency code, confirming our hypothesis that long latency code attends to more fine details of the faces, optimizing it for face discrimination. Moreover, when we try to cross-predict from one time window to another, the performance dropped hugely, again showing that the short and long latency code are totally different from each other. And with that, I would like to stop here and summarize my talk. We've shown that at short latency, face cells used a general purpose code for faces and objects, optimized for face detection. At long latency, face cells used a specialized code optimized for face discrimination. And this code change does not happen for objects. We think these findings answer several key questions in vision. What is a face? A face patch considers a stimulus a face if the stimulus triggers a state change. Face cells achieve specialization for faces over time through a rapid concerted change in the neural code. Secondly, IT neurons don't have fixed tuning functions. And we reveal a new form of neural computation for object recognition, which hopefully will generalize to other areas in the IT cortex. And with that, I would like to thank my advisor, Doris Tsao, my colleagues and collaborators, Dasheng, Janis, Frank, and Shi, and also to postdocs, Kritika and Nate in our lab, who have helped me tremendously, and of course, all the funding sources. Thank you.
Go ahead, keep speaking.
Working. I can just. Hello. Is that working? So yeah, super amazing, cool. I was really convinced by the Bao and Tsao paper showing that the representation for objects and faces was all part of one representation, and it mapped anatomically across the inferotemporal cortex. Mm-hmm. And now I have to rethink. I don't know at all how to fit what you just said in with that thing, which seemed to be obviously true to me. So maybe you can help me.
Oh, yeah. I think the key question in our finding is that the cells can actually multiplex domain specificity across time, right? So in the early time, it's doing this face detection, which is using a general code, which is exactly as shown in the paper from Bao and Tsao in 2020. Yes. So there's a different temporal contamination when you average through a long time window. Because in the short latency, the response amplitude is higher. So that's like kind of dominating the response. But now we're not looking at the amplitude, but the tuning, like the relative difference between faces coding. Yeah.
Okay, I think we're going to have to wrap up the session. Sorry, you can catch the speaker later for more questions, because we have a coffee break coming up now, and session 2 starts at 11 o'clock. Thanks, everybody.